So continuing with the fourth screencast for chemical and biological risk or human health risk, we are now turning to the topic of toxicology. Toxicology is the study of chemical risks. So first I will define the 5 categories of chemical risks and give some examples, and then we'll look at how scientists study these risks. So the first category of chemical risk is neurotoxin. Neuro is, you see the beginning of the word is neuro as in neurology or nervous system or neuron. So neurotoxins are chemicals that disrupt the nervous system. The nervous system consists of the brain, the spinal cord, and any of the peripheral nerves that leading all the way out. So common neurotoxins include insecticides such as DDT, and heavy metals such as lead, mercury, cadmium, and all of these others that we've discussed. So those are neurotoxins. The second chemical risk category is carcinogens, and that is a chemical or chemicals that cause cancer by inflicting damage to cells. The cells might be a scarring of tissue, or it might be damage to the DNA controlling the reproduction of certain cells. And the most commonly known and problematic carcinogens include tobacco smoke, asbestos, and radon. The third category of chemical risk is a teratogen. A teratogen is a chemical that interferes with the normal development of embryos or fetuses. So one of the most common teratogens is alcohol. So if a mom drinks alcohol, especially excessive amounts of alcohol, while pregnant, that will affect the development of the baby, including causing great difficulties with IQ and some physical abnormalities as well. Certain medications are also teratogens. One of the most commonly known teratogen medication is thalidomide. Thalidomide, you might have heard that word if you remember the Billy Joel song, We Didn't Start the Fire. He talks about thalidomide. And thalidomide was given to women in the 1950s and early 1960s to treat morning sickness. The problem is it actually caused great damage to the fetus. And the teratogen thalidomide was a medication that was under review by the FDA at the time, but the FDA did not take action, either approving it or not allowing it onto the market. And it was prescribed what we call off-label. And so on the one hand, you had the FDA saying, well, we never approved it, it's not our fault. On the other hand, they had a mountain of data on their desk that indicated that this medication was a teratogen and it should have been taken off the market and people should have been warned. So nowadays, though, the FDA has much more stringent criteria for reviewing and the amount of time that they're allowed to review a medication, so that hasn't been as much of a problem anymore. So that was the first 3 of 5 chemical risks: neurotoxins, carcinogens, teratogens, and we'll move on to 2 more. Next we have allergens, just like the word allergic. An allergen is a chemical that causes an allergic reaction. And sometimes these allergic reactions can be very serious and even fatal. Common allergens include nuts, bee stings, medications such as penicillin, and even animal dander. And the final category of chemical risk are endocrine disruptors. So first let me discuss what that means, what a hormone is and what the endocrine system is. A hormone is any chemical that's made in one part of the body and then travels to another part of the body, to another organ, to act on that organ. And the endocrine system includes a whole series of systems where hormones are produced and act in different parts of the body. And common parts of the endocrine systems are reproductive hormones, and that would be just one part of an endocrine system. So endocrine disruptors are chemicals that interfere with the normal functioning of hormones in an animal's body. They might disrupt the flow, they might mimic the normal hormone and interfere with its action in that way. And one of the biggest problems is that hormonal waste, particularly that from birth control. So a woman takes birth control pills, and then some of that hormone is eliminated through urine. And that hormonal waste may have an effect on fish and amphibians, and may even affect humans. For fish and amphibians, it particularly affects their reproductive systems, making some of them hermaphrodites, so they're both male and female when they normally would not be. All right, next we're going to turn to some of the way that scientists study these chemical risks. So scientists want to know at what concentration is a chemical harmful. Maybe it's okay in a lower dose or lower exposure, but then becomes harmful in higher concentrations. So in order to determine that, two types of studies are done. Two types of dose response studies are done. The first one, LD50, stands for lethal dose 50. That means you have the dose at which 50% of the tested individuals die. So if you're testing, let's say, a group of 100 mice, and you gradually increase the dose until a dose at which 50 of those 100 mice die, then the LD50 for that particular chemical has been reached. So the LD50 can determine a relative toxicity, but it varies greatly among species for a given chemical. So what might affect mice may be more harmful to amphibians or birds or any other species that you can think of. So sometimes, in addition to lethal dose, scientists measure something called an effective dose. An effective dose is a misleading term. You might think that means a positive thing, but it means when a negative effect is shown in 50% of the tested animals. So an ED50, or an effective dose 50, is the dose at which 50% of the tested animals display a harmful but not lethal effect. So the harmful but not lethal effect might be an inability to reproduce, or an inability to issue a bird song, or an inability to walk or swim. So all of those things would be an effective dose, but not a lethal dose. Okay, so here we are looking at a hypothetical graph of a hypothetical chemical and its lethal dose 50. So you can see that going across the x-axis, we have the dose increasing from 0 to 4, 6, 8 to 10 units. And going on the y-axis, we have the percent of mortality, or the percent of test organisms that die. And you can see that it's an S-shaped curve. So at lower doses, the tested organisms are not dying. And then there's an increase and eventually a flattening of the curve until 100% death rate occurs. So if we follow across at where the 50% intersects the curve and follow down to the x-axis, you can see that the lethal dose 50 for this hypothetical chemical is at about 5.5 units. Once the lethal dose or effective dose is known, then regulatory agencies need to determine at what level is it safe, what dose is safe to have out in the environment, and what level is safe for humans, because we're not going to do lethal dose studies on humans, of course. So what happens is they take the lethal dose 50, let's say in this case 5.5, and take that and divide it by 10, or take 10% of that. So if the lethal dose 50 is 5.5, then dividing that by 10, you get 0.55, which would be about here on the curve. And that level would be considered safe for being present in the environment. Now, in terms of humans, it's a little more strict. It's either 100 or 1,000th of the lethal dose 50. So if your lethal dose 50 is 5.5 here, then you're getting down to 0.055. So that would be even further down the curve here. And if you divided it by 1,000, you'd be even closer to this point here. So it gives a general reassurance that we're in this part of the curve and therefore most likely not to be harmful. But again, we're sort of pulling that number a little bit out of a hat, because just because it's not causing at that dose death, it does not mean that there aren't going to be other harmful effects or cumulative effects from having multiple exposures to that particular chemical. So let's take a look at that next. So in real life, life is not a dose response study, and we need to look at some of the other factors that make something more or less toxic. And these aren't really totally spelled out in your book, so make sure you take a good look at this slide. So the first factor is exposure dose, so that would be the lethal dose type of calculation, and the length of time of the exposure. So is it continual exposure over and over again to that same chemical, or is it just a one-time deal for a period of, say, 1 minute? A second factor is individual sensitivity. Younger people, children, and older people, senior citizens, tend to be more sensitive to chemicals. For the younger children, it's because of their high metabolism. And for the older individual, it's because some of their responses are lessened. So an example here is when I was in college and living at home, and it was the summertime, and we had the windows open, and I was in the kitchen with my mom, and all of a sudden she had trouble breathing, and she never had asthma or anything like that before. So we went to the ER, and the ER was full of people who were having trouble breathing. And it turned out that they had sprayed for mosquitoes and right over the neighborhood and just had been doused with this stuff. And some people were sensitive to that and got sick. I was in the same room at the same time, and I had no issue with it whatsoever. So that's individual sensitivity. A third factor is, the third and fourth factors go hand in hand. If something is soluble in water and if something is soluble in fat. So we know that sometimes if it's soluble in water or fat, the chemical will bioaccumulate within an individual and then biomagnify as you go up the food chain. So those two taken together, bioaccumulation and biomagnification, may make something more toxic. The age of the individual, I sort of addressed that when I was talking about individual sensitivity, but the age of an individual can make a difference. Persistence, which has to do, again, with that first item, length of time. If a chemical is more persistent, then it will be in the environment longer. Synergistic interaction means that two risks together cause more harm than either one alone. And a great example is smoking and asbestos. If an individual smokes and they have a risk of, say, 1, and if they are exposed to asbestos and they have a risk factor of 1 for that, but they both smoke and are exposed to asbestos, they don't have a risk factor of 2. They would have a risk factor of 5 because there's a synergistic interaction between the tobacco smoke exposure and the asbestos exposure. Okay, that's a wrap for this video, and we'll move on to the next one.